Hi there. Welcome to the From Lab to Launch podcast by Qualio, where we share inspiring stories from the people on the front lines of life sciences. Tune in and leave inspired to bring your life-saving products to the world.
Hello, everyone, and welcome to From Lab to Launch by Qualio. I'm Kelly, your host, and we're glad you're tuning in today. Before we jump in, just a reminder to please rate the show and share it with any of your science nerd friends. We know you have some. Also, check out the show notes if you have a story or a product you'd like to share with us. We love hearing from you. Today, we're very excited to have Dr. Marcus Conant, Chief Medical Officer at American Gene Technologies. American Gene Technologies is a pioneering biotech company headquartered in Rockville, Maryland. They're using gene therapy to cure humanity's most deadly diseases. AGT is creating the foundation for a fundamentally new approach to medicine. Much like Apple's iOS platform for the iPhone, AGT is building a gene therapy platform so other scientists can build on its approach to develop new cures, much like software developers creating apps for the iPhone. This will significantly cut the time and cost of developing new drugs for a variety of diseases. We're really looking forward to chatting with Dr. Conant today about his experiences in the industry and journey to where he is now. So let's bring him in. Good morning, Mark. Thanks for joining.
Thank you, Colin.
To get us started, tell us about your background.
It's a long story. I trained, believe it or not, as a dermatologist and was on the faculty at the University of California in San Francisco as a practicing dermatologist when the AIDS epidemic began in 1981. And the first cases that we saw were Kaposi sarcoma, which is a skin disease. It's a multifocal malignancy. It can occur internally, but it appears primarily on the skin. And so as a dermatologist at an academic center in San Francisco, which had a huge gay population, it was obvious that we would start seeing large numbers of those patients. So I started a clinic at the university. That clinic grew to huge size. I started seeing patients in my private practice, ended up building the largest private AIDS practice in the country, where we were caring for some 3,000 HIV positive men. This was in the period when if you caught HIV, you had a 96% chance of dying. And so we lost a huge number of those patients. But by 1996, we had highly active antiretroviral therapy. And so suddenly it was like the Lazarus syndrome. Patients were coming back from the dead, recovering, their CD4 counts were going back up, and they were doing great. So I practiced dermatology. Yeah, it was a miracle. It was a miracle. You know, it was truly a miracle. And it's sad that so much of that history has really been lost because we lost a whole generation of very bright, young gay men who had moved to San Francisco or New York or other metropolitan areas to get away from the prejudice that they were experiencing in the towns where they grew up. They were obviously bright, capable, able to get a job, find a boyfriend, settle down, and then suddenly at age 32, with a career ahead of them, everything working out, suddenly they're dying of a disease for which we not only had no cure, we didn't even know what caused it. And think of how frightening that must have been when you know your doctors don't even know what you got. But anyway, that was the period. It was terrifying. It was truly terrifying. And it was terrifying for us treating the patients because we knew we didn't know what we were doing, you know. But anyway, that was all in the 1980s. Now we're 42 years after that. And still, there are many things about the AIDS epidemic that we don't understand. We know now what causes the disease, but we can suppress that virus with drugs, but we can't eliminate that virus from the body. And this will come up over and over in our discussion today. But one of the reasons we can't eliminate it is the virus literally becomes a gene. When you catch HIV, you're catching a gene, and it's integrating into your genome, and it's there for the rest of your life. We can suppress it with drugs, but we can't go in there and take that gene out. And so that's now the challenge is how can we suppress that gene? How can we keep that gene from manifesting itself so that the patient gets a functional cure? And we can talk as we go later about other conditions where that seems to happen, or doesn't seem to happen, it does happen in patients.
Absolutely. Well, it seems that's one of the innate evolutionary, amazing and horrible traits of viruses in general. Right? Where, you catch one, you've got it. Some just happen to be nastier than others, it sounds like.
That's right. And we think that somewhere about 10% of your genome are viruses that our ancestors have caught in the past.
Interesting. Well, I was going to veer off in the direction of the Kaposi Sarcoma Research Foundation, but I feel like we should keep on this viral thread a little bit. So, as far as what AGT is doing and how the viruses and all of that sort of ties together, I assume that's still an area of active research and something you guys are trying to use to inform some of your tech decisions.
Right. Well, yes. And let me give you a little background. About 12 years ago, the founder of this company, Jeff Galvin, who came, by the way, from a computer background, he worked with Apple for many years. It suddenly occurred to him that, hey, wait a minute, if many of these diseases are in fact genetic problems, perhaps we can treat these diseases by either removing that gene or in some way blocking that gene so that the gene is no longer operational. Well, for example, Kaposi sarcoma is caused by a herpes virus, which integrates into your cells. If your immune system's normal, your immune system suppresses Kaposi sarcoma. You don't have it. But if you catch a virus like HIV, which destroys your immune system, the Kaposi sarcoma manifests itself. So the virus is there, and it only comes out when you're immunosuppressed. And so you take an AIDS patient who has Kaposi sarcoma covering his entire body, you put him on highly active antiretroviral therapy, you suppress the HIV virus, his immune system returns, and what happens? The KS all goes away. And we saw that in hundreds of patients. It literally fades away in front of your eyes. And so what's happening there? The patient is still infected with HHV8. He still has the ability to produce Kaposi sarcoma. But if you can some way suppress that gene, then you can get a functional cure. And think of it, many diseases are because you have either caught an inappropriate gene and it's manifesting itself, or you don't have a gene that you need. And if we could put that gene in the body, you've taken care of the disease. And so that was what occurred to Jeff is, wait a minute, if we can now using some of the technology that we now have, we can go into the body and either suppress genes or add genes that aren't there, we can get a functional cure for these diseases. And one of the first diseases to attack, of course, was HIV. The disease is, as I said earlier, you're catching a gene. How can we suppress that gene? And so that's what we're doing. We're taking a lentivirus, and I'll explain it in a little more detail, but we're taking a lentivirus, we're putting constructs in there that suppress the HIV virus, and we're putting those cells back in the body, and we're suppressing HIV so that it can't replicate.
Interesting. And if it's not actively replicating, perhaps it can't be spread?
Correct. And we know that's from treatment with pharmaceutical medications, we know that's true. If you take pills and get your viral load below the level of detection for a period of 6 months, you are no longer infectious. U equals U. You are no longer infectious. And so if with our gene therapy, we can suppress the virus down to very low levels, hopefully to get the patient undetectable, the patient will no longer transmit and will, of course, not manifest any complications of HIV infection.
Interesting. I'm not sure about what the side effect profile looks like of the retrovirals and those drugs that they have to take, but I would assume that in this genetic model, you're dealing with less of those kinds of pharmacologic and side effect type activities, right?
And we have a little data, not only
good.
from our studies, but from other studies as well. First, about the pharmaceutical drugs. It is true that early on, the side effects were horrendous. I can remember starting the first patients on AZT, which was the first drug that we had that was effective, and that was in 1988, I believe it was. And that was an awful medication. You had to take it every 4 hours or so. You had to take it 4 or 5 times a day. You had to make very large doses. Most patients, it's upset their stomach. It caused all sorts of gastrointestinal complications. It made patients sicker than hell. But it did the important thing. It was proof of principle. It proved that there was a way to suppress the HIV virus with a drug. And so that starts the process. Aha, we know now that we can do it. Now let's find drugs that do it better and that have fewer side effects. And so that's what the American pharmaceutical industry did for the next 30 years, was they have now developed drugs that have hardly any side effects. It's only one pill once a day. It's usually a combination of 2 or 3 drugs in that combination because this virus mutates very frequently. And you can suppress the virus in patients that take the medication regularly, in essentially all patients. And studies show that those patients live essentially a normal life. They can live as long as they would have lived had they not contracted HIV. The problem is that they have to remember to take the drug every day. And that sounds really easy. You think, well, you've got a fatal disease that can kill you. Any idiot can take the drug every day. But life gets in the way. And so suddenly you don't have enough money to buy the drug this month. Or the car broke down and you can't get to the pharmacy. Or you happen to live in a rural area and you're too far away from the pharmacy. Or you're a mother of 3 kids and you're HIV positive and one of the kids is sick. And you don't have time to get to the pharmacy. All of those problems come up. And so compliance is not 100%. If you could put cells in the body which will suppress the virus and they're there for the patient's life, you no longer have any of those problems. And something else you accomplish. The patient that has to take a pill is reminded every day that he's HIV positive.
Yeah.
Every day you go there and you have to take that pill. Yeah, I'm still HIV positive. And all the things I wanted in life have been changed. I now have to see a doctor 3 or 4 times a year. I have to tell my sexual partner that I'm HIV positive. Is there a chance I could transmit this to someone that I'm intimate with? On and on and on, all of these questions. When I go to the pharmacy, do they look at me different because I'm HIV positive? All the human concerns. If we could do it genetically, if we could take the patient's cells, modify them so that they can't get infected with HIV and put them back in the body, that patient then has a functional cure which would suppress the virus for the rest of the patient's life.
That would be an incredible life change for them.
That's right. And for all of us.
So where is AGT in the process then of getting that developed?
It's always too slow, Kelly. It never goes as fast as you want it.
It is always too slow. I agree.
Right.
Yep.
Jeff Galvin's been working on this for about 12 years. I joined the company about 1.5 years ago. And we've got some good data. We have put first, briefly, let me tell you how you do it. You take the patient and you leukapheresis them. You take their own cells out. So just like you're taking blood for transfusions, you take the blood out, but you put the blood back in and you just save the white blood cells. You take those white blood cells and you grow them up from hundreds into billions. And you take a lentivirus vector. A lentivirus looks very much like the HIV virus, but you've rendered it so that it's no longer infectious. It can't spread from one cell to another, but it can infect those cells. And we have genetic conscripts in there that block CCR5, one of the necessary molecules for the HIV virus to enter a cell. So you block CCR5, and you put that, you take that lentivirus vector, you put it into those cells, and so now those cells are protected against being infected with HIV. You infuse those cells back into the patient, and then hopefully the patient no longer can, those cells will kill HIV, but will no longer be killed by the virus. We did 7 patients. In all of the 7 patients, 3 things. There were no adverse events. In other words, the patients didn't get seriously ill. The worst that we saw were what you see with a normal transfusion. People get headaches, people get chills from a transfusion. That's all we saw. There were no serious adverse events that need to be reported to the IRB. That's great. That's exactly right.
That's amazing.
But then there are 2 other things that could happen. One is that the body could say, wait a minute, you've messed up my cells. That's no longer me. That looks different. That's non-self. And I'm going to reject those cells. But that didn't happen. So we saw no rejection of the cell. But there's even something else. Sometimes when you do a graft, engraftment, the graft rejects the host. The cells could have said, you don't look like me, I'm going to reject you. And that's called graft versus host disease. And that didn't happen. And so the things that we really wanted, was it safe? Yes, looks like it. What did it reject the patient? No. Did the patient reject the cells? No. And the other thing we know is those cells persisted. Now, they didn't persist at the levels we want because they need antigenic stimulation. Those cells over time will slowly go away unless they're seeing virus. But these patients were still on their antiretroviral medication. And as I've told you, if you're taking your medication, you're not infectious. There's no very little virus in your bloodstream. And so those cells that we put in weren't seeing virus. And so over time, we know that they decreased in number. Now, when we took the patients off of their antiretroviral therapy, it looks as if those cells are active. But how active they are and how many were still persisting, we don't know at this point.
Gotcha. Okay. Yeah, that's what a fascinating trial to design.
Well,
lots of endpoints, lots of measurements along the way, and
lots of unknowns.
Yeah, definitely.
Things that you don't know is, for example, when you put the cells back in, how long do you wait before you take the patients off of their antiretroviral therapy? Do you do it right away? Do you wait a month? Maybe if you don't wait long enough, the cells don't engraft and they'll be rejected. All these things we don't know. Of course, now, as we do the studies, we will learn those things. And so we're preparing right now to do another study beginning hopefully next year in which we will answer some of those questions.
Good deal. And these are all in human, or are you doing some in primate research? Or
Well, this is one of the bizarre things about HIV. The only animal you can infect with the human immunodeficiency virus is humans. Early in the epidemic, Jay Levy, where I trained in San Francisco, and others tried to infect other animals, dogs, cats, rats, the things that you normally do. We were unsuccessful in infecting any other animal with HIV. And so the studies have to be done in humans. And of course, that requires humans to be subjects to volunteer for clinical trials, which requires a huge commitment. Not only a commitment to engage in a trial where we clearly don't know all the answers, but the time that it's involved. For example, in the new trial, the patients will be seen probably once a week for some 30 weeks. Can you imagine the commitment of going in once a week to see a doctor and have blood drawn for a period of 30 weeks? That's a huge commitment.
That is a huge commitment.
And yet, that's right. And yet we found in this study, there are people who care enough about this disease, who've seen enough people get sick from the disease, seen enough people impacted by HIV, know the history of how many, we lost 800,000 Americans to this disease, that we right now have 1,200,000 infected Americans, that we need to find an answer for this disease. And even if it's taken 42 years, the answer's out there. We know we can cure this disease, because remember, with Timothy Brown, the Berlin patient, he developed a form of cancer. The doctors caring for him said, look, when we do a bone marrow transplant, let's don't just do a bone marrow to cure the cancer. Let's do a bone marrow transplant that has this CCR5 mutation, which is the very thing we're doing, that will block HIV replication and put those cells back in. And they did that. And what do you know? They cured him. And he lived another 10 years or so before his cancer returned, and he died of his cancer. But during that period of time, they cured his HIV. And that's been done now in 5 other patients. So we know we can cure this disease. It's just, how can we do it without having to do a bone marrow transplantation?
Right. Yeah, that's awfully invasive. Wow. Well, you're clearly passionate about this. You're a physician with a very holistic perspective. What does that mean to you in this case? What does that mean to you in general?
Well, I was a dermatologist practicing at a university. I'd been practicing 15 years or so when HIV came along. And suddenly I saw my neighbors, my friends, my colleagues, the people I went to the opera with, dying of this disease. It's life-changing because at first you think, I'm just going to be treating these people and it'll be over in a few weeks. And then it's a few months. And now it's 42 years. And most of those people aren't there anymore, but I'm still here. And so as long as I'm here, I can continue this fight for them and for myself and for everyone else. We can cure this disease, but we've got to keep doing the work.
Definitely. Is there a particular impactful story or interesting story along the way that you could share?
Well, there are lots of them.
Lots of them, I'm sure. Is there one that stands out, maybe?
Well, one of them that I often tell is I had a very good friend, a gay guy, who I had known for maybe 10 years. And and this was before the first case was seen in 1981 of AIDS. They brought Al to my office because he had a splitting headache. And I couldn't diagnose him. It was obviously needed to be hospitalized. He didn't have insurance for private insurance, but he was a veteran. And so we admitted him to the VA hospital that day. He went into coma that night, and 6 months later, he died. Okay? At autopsy, he had tuberculosis of the brain, extremely rare. I mean, I've been in practice now 60 years. I've never seen a case of tuberculosis of the brain.
I didn't know you could get it in the brain.
Well, you see, what it is a very rare opportunistic infection. His boyfriend at the time was an IV drug user from New York. Okay, and this was 4 years before the AIDS epidemic began. In retrospect, he had AIDS. We didn't know it at the time. What I'm trying to point out is we were seeing the disease, but we were not recognizing what we were seeing years and years before the epidemic really began. And in all probability, he contracted that disease from his IV drug-using boyfriend. It was immunosuppressed. The tuberculosis manifested itself in his brain and killed him, unfortunately. So those kind of things, throughout the epidemic, one really strange event after another.
That is, wow. Yeah, I sorry, my brain is spinning a little bit with how do you even begin to connect those dots? But definitely, I think in retrospect now, seeing, and I think probably today, although I would say certainly, COVID is nowhere near the impact seems to be more of a flash in the pan compared to something like HIV. But yeah, I think probably for our general listeners, just thinking about some of those parallels with seeing some different kinds of infections or different presentations and then realizing later that you've connected all those dots together. That's
but and to your point, it makes 2 points that I think are important. One, it points out, and COVID made the point very clearly, that if we had better surveillance so that we recognized infectious diseases as they appeared, if we had a worldwide system of recognizing diseases like COVID or like AIDS early on, we could hopefully prevent the 10 million people that had died worldwide from COVID.
Right.
But the other point that I keep trying to make is, remember, HIV, for the first 10 years of the disease, killed 96% of the people that got the disease.
Yeah.
If a young man came to me and I told him that he was infected with HIV, what I was telling him is, you're going to die. And no one wants to hear that. Okay.
Right.
COVID only kills about 2% of the people that get it.
Yeah.
Okay. What if COVID had killed 20% of the people that got it? Society would have collapsed.
Yeah, that's a very different scenario.
It's a huge difference, right? And are we really prepared for that to happen? And then you say, well, it's never going to happen again. The Spanish flu epidemic was exactly 100 years ago in 1918. And here this disease, of course, has killed more people, but there are more people living today than there were then. But, the same thing happened 100 years ago. But in the interim, we've had Zika, we've had Ebola, the list, we've had AIDS, the list goes on and on and on. And the chances are we will have another major epidemic, which not only will shut down society like we've seen, but may in fact kill more people than COVID and will totally disrupt our ability to function as a society.
Yeah, definitely. Definitely. Oh, goodness. Well, if you could go back and tell yourself something at the start of your career, what would it be?
Well, I've thought about that a lot because, when you get to be an old man, you start thinking about what would I have done differently, in my life. Looking back, I probably would have become a virologist rather than a practicing physician. I mean, I find that incredibly exciting. But who knows? I've been truly blessed at being on the front line of lots of things, on the front line of HIV happening in San Francisco 42 years ago, now on the front line of gene therapy in a way to try to cure this disease. So I really can't complain about how life should be different because it's been pretty good.
Definitely. Well, and some might argue that your broader focus in medicine perhaps brings a different perspective than specializing just in virology, for example.
Perhaps.
Perhaps. Awesome. Well, last question, my fun question of the day. I always like to ask, if I walked into a bookstore like Barnes and Noble, where would I find you? What section?
Oh, that's a very good question. Probably history. I mean, if you ask me what I'm reading right now, I'm reading Brunelleschi's Dome about the guy who built the dome over the cathedral in Florence. So outside of medicine, my interests tend to be historical.
Historical. Interesting. Well, and they do play together so nicely. There's so much medicine involved in all of history.
That's exactly right. And so much history of medicine.
Definitely. Well, thank you so much for your time today. I really appreciate it. This has been an interesting discussion. Where can folks go to connect with you or follow along with the therapies?
American Gene Therapy. They're on the web with a huge presence. If you just go to your computer, check for American Gene Therapy, you'll find us.
Awesome. Thank you so much for your time. Have a wonderful rest of your day, Mark.
Thanks, Kelly.
Thank you for listening to this week's episode of From Lab to Launch, brought to you by Qualio. If you like what you've heard, please subscribe and give the show a positive review. It really helps us out. For more information about Qualio, our guest today, or to be a guest on a future episode, please refer to the show notes. Until next time.